The next speaker is Khanna from the National Cancer Institute. We'll be talking about advantages and experiences with trials that include animal patients.
Thank you much, Dr. Kasten, and thank you very much to the IOM and to the leadership from CSU to bring this workshop together. So certainly, as Dr. Kasten suggested, one of the primary goals of the meeting is to identify gaps in the delivery of this opportunity. But I want to point out a reflection of the advances that we've made in the field, and that is that previously I would have to worry about where I was going to talk relative to Matthew Breen, because Matthew Breen would always show the same pictures of the dogs that look like humans. And that was my concern when I looked at the agenda. But now I have to worry about the center director at Duke and others. So that alone is a reflection of where we've come. So I do think we need to identify gaps, but there has been progress. So I want to take a quick sec to outline my disclosures, but they also, I think, help present my perspectives on this overriding opportunity. And the first is that I work in the area of cancer biology and translation, currently as a consultant to the Pediatric Oncology Branch and the Center for Cancer Research at the National Cancer Institute. But that work has mostly been involved in understanding why cancer spread, and was reflected in my questions to Dr. Hellman. The second is through my work in comparative oncology. Sorry, don't seem to have a pointer working. Patrick, do I use the screen or? Okay. No, as a pointer, sorry. Evidently not. Oh, the blue is a pointer? Oh, right. Right. It's not a pen. Okay. So the second is my work through the comparative oncology program, which has been introduced, and I'll give you some additional senses of where we work there. The next one that's important is my work as a clinical veterinary oncologist, where through our practices that are located in this area, we see over 4,000 new cancer diagnoses a year, and we have these discussions with pet owners about what's the best option for them in terms of pursuing treatment for their pet, whether that's a clinical trial or conventional chemotherapy, for example. And then in animal health, my work involves consulting work through animal clinical investigation, which is a CRO involved in cancer drug development primarily in animal health. And then and then also as a consultant with Abbott Animal Health and as an advisor to Aratana. So I have been involved both in the human side of animal-related clinical studies as well as animal health-related studies. And then as a nod to my future disclosure, a relationship that is growing with TGen, where I expect to join and work in cancer biology and animal health, both within TGen itself and a new animal health affiliate of TGen. So I think that gives you a sense of where my perspectives have come from. And there is, again, the slide that was stolen from me. Okay, well, there you go. Again, I think I'll take that as an acknowledgement of progress in our field. And Dr. Hellman and I did discuss sort of the strategic value of sharing some slides, and I think that our perspectives are different enough that we will likely say different things about the same slides, and I hope that we can use that as an opportunity. And so listed here are the advantages and opportunities of the dog as a model, and we look at that as compared to the human as a model. So I make that point because I'm not afraid of describing the human in a human clinical trial as a model of disease, nor as a dog in a clinical trial as a model of disease. So these are large animals, and that historically has been a big part of the reason that we've used them as models. But a long list of potential values, including where we've looked, the strong genetic similarities to the human. We haven't looked as far as we need to, but where we've looked, there are very strong similarities. They are naturally occurring. As Bev pointed out, the issue of sort of syngeneic relationships with tumor and microenvironment are all consistent in these systems. And if you look at, so if I ask the 10 drugs that work, or ask the medical oncologist in the room, what are the 10 drugs that work in lymphoma? those same 10 drugs work in our lymphoma patients in dogs, and the 3 that don't work in our patients don't work in humans. So as a post hoc assessment, you could say there's some value to predictivity, but I do not like the term predictivity in model systems, because the primary role of the model is to answer a question, and the responsibility is of the scientist to ask the right question. It's not really the model to deliver the right answer. It's you to ask the right question. And I think that these 3 are in red because they capture the essence of the problem of cancer in human patients. And I believe that because of the cell-to-cell heterogeneity that exists in dog cancers, which doesn't really exist in the mouse models that I use, these natural problems which we see in dogs, which is recurrence, resistance, and metastasis, emerge. And that is, as I said, the essence of the problem of cancer in human patients. So as a cancer biologist, this heterogeneity drives the real problems that have needs that we all, I think, intend to deliver on. So this slide just shows you that beyond the cartoon, there's at least a reason to ask the question about whether the dog with cancer can help us understand drug responses in humans. And this is a microarray experiment that Melissa Paoloni in the lab did, comparing human osteosarcoma and dog osteosarcoma. And by cluster analysis, you can see that across the thousands of orthologs, you can't segregate the dog and the human. You see normal tissues clearly being different than tumor tissues, but the dog and the human do not segregate differently. And where this has been looked at in other dog cancers, in lymphoma, bladder cancer by Dr. Knapp and others, the same sorts of things have been shown. So there is at least a reason to ask this question, and I think that's why we're here. And the other point that I'd like to make is that I don't think the discussion today or tomorrow is about using the dog as the approach, but rather to add the dog as a model to the other model systems that are used, so that the totality of information that's available to us as we ask questions is enhanced. But not to suggest that this is necessarily better than this, but I think this asks or allows us to ask questions that we can't ask in these models or in these models. And I think that ability to ask questions is a theme that I'll return to through my brief presentation. So I think that the conventional drug development path is flawed because of it being unilinear, and because these people who study preclinical drugs really don't have an influence in helping make decisions as the process advances. And I think Lee brought that up very well, and leads to the idea that, sorry, I'll just jump ahead, that there could be questions that we could ask in the dog with cancer that helps design the best phase 1 human trial, and maybe more importantly, the best phase 2 and the best phase 3 trial. And we have gone through the questions, but they're simple questions about the dose, the regimen, the schedule, the biomarkers, which targets we should really address in phase 2. And so I think it's more about how these important models can be added to these other models as we design better human clinical trials, so as to address this issue of late attrition. This is certainly not a new idea. And from this review article that we published in 2008, work by Rainer Storb and others in the 60s, developed preparatory regimens for lymphoma in dogs with lymphoma. And these were for bone marrow transplantation. And over the years, a number of important studies have been done and have led us to this point in time where I think we would call it the integrated opportunity. And what this means to me is we can sit down with people who develop drugs and we can say, what are the questions that you're having a difficult time answering in the mouse or in the human? And then we can say, well, we think we can answer these questions, or we don't think we can answer these questions. And so we sit down and we, in a discussion, decide if the dog is the right way to answer these questions. And so I think that's where the opportunity stands now, largely as a result of the genome in the dog, advances in genomic techniques, and the availability of pieces of infrastructure and relationships that are all represented in the room. So if you're sort of pushed to ask, where is the value of the dog, we came up with one way to sort of quantify the value. And so the red line is the conventional drug development path, and the blue line is an integrated or comparative drug development path. And if you look at the distribution of drugs across the phases of approval, something very subtle by blunting this steep portion of the curve between phase 1 and phase 2 through the comparative approach. So if we were to be successful simply in finding failures early so as to eliminate some phase 2 trials or to design better phase 2 trials, which is what was represented here, the opportunity looks like this. And that's that we save money in the comparison of these two approaches. The number of drugs that is approved doesn't really change, but the number of drugs that fail in phase 3 and phase 2 changes. And that's where that savings of money and the savings in patient outcomes is seen. So I think, again, that's a very, very small and very focused area that we could look at for this opportunity, including comparative oncology. But the magnitude of that subtle difference between phase 1 and phase 2, sort of focusing on improving phase 2 designs, is, I think, quite large. And another way to sort of look at the value is to look at the approach to sort of take a reduced risk as we are moving from phase 2, for example, to phase 3. And that reduction in risk is really through answering questions that we don't currently answer well. And by doing so, we shift the dynamics of these relationships between risk and reward, and we create less risk as we move towards the higher sort of risky drug development steps. And so these are very subtle changes that occur by answering good questions. And I think as we decide on where and when to use these models and develop these answers, I think there are strategic windows during drug development in various pharmaceutical companies where not a lot happens, and by being prepared, you have the time to answer these questions and decide if the result of that answer sufficiently reduces risk because of the unknown or by reducing cost. So there's a balance that I think by being strategically thoughtful, you can achieve a valuable sort of decision on where to include these questions. And I think there are all these valuable gaps that occur in every drug development path, and they're all slightly different in each company, but they do exist so that when you're thoughtful, you don't necessarily have to delay an existing drug development process. So in terms of the opportunity, we have a national infrastructure through the NCI called the COTC, Comparative Oncology Trials Consortium. And this is the consortium now led by Dr. Amy LeBlanc and Christina Mazco, and you'll hear from Amy later. So that's one of the primary efforts of the program at the NCI. We have an opportunity within the NCI and the community to develop approaches to ask questions about why drugs work, but more importantly, why they don't work in these trials, and then to advocate for when the trial should be done and when the dog is perhaps not the best way to answer these questions. The approach through this consortium has been successful based on the number of studies that have been conducted, completed, and then have moved on to publication. So we do have a mechanism, I think, to deliver these multi-institutional studies now. Within the studies, the data is collected contemporaneously through electronic reporting systems, so the data can be viewed and reviewed in real time, which is important to our pharmaceutical partners. We have an understanding from a meeting that was convened in 2008 about how these data should be reported to regulatory bodies, and that's with existing guidance. And in this meeting in 2008, many of us came together to discuss whether there was sufficient clarity on how these data should be reported during the conduct of the study. And I think the outcome of this meeting was that there was clarity both in the pre-IND and post-IND settings. And if you look briefly at how the studies are now developed, they are developed through the discussion of what questions are not being answered and asked. And I think that's an important sort of starting point. And then we then decide, can we do it with what we know? And then we design the study. And they're very rapidly, sort of in a sequence, very rapidly delivered designs of studies. And sort of to give you a sense, coming back to Dr. Hellman's discussion, these are not complicated clinical trials. We're primarily taking advantage of the fact that we can dose dogs with a variety of doses of drugs, do PK in those studies, collect tumor and normal tissue before and after exposure to drug. And as you hear from Dr. Choyke and Dr. LeBlanc, you can ask questions about the validity or the applicability about various imaging approaches, which could be biomarkers, validate those imaging approaches against tissues collected in these studies. And so this is the sequence of most of these study designs. Does the drug work? Can you safely deliver a dose? Can you achieve a therapeutic exposure that's relevant? And do good things happen to biological targets and to biological surrogates? So I think that summarizes the general design of many of these studies and highlights the opportunity to ask questions in these studies that are difficult to ask in human clinical trials. Dr. Breen will talk about this opportunity, and I think it's fair to say that we don't yet know enough about the genomic biology of these cancers. So there's not an in silico library of the genetic characteristics of these cancers. But we do have the tissues collected, and Dr. Breen will address that. And so these questions can now be asked in a very rapid manner. And these tissues are collected within a consortium called the Canine Comparative Oncology and Genomics Consortium. These tissues are available at request, scientific request. They're very carefully collected, and the quality of these tissues has been shown to be extraordinary at every level, nucleic acid and histological evaluation. So the tissues exist for such biological questions. In addition, through the trials mechanism, we've collected through the leadership at CSU and Dr. Tham and Lana, a mechanism to ask biological questions. So coming back to Dr. Hellman's point about biomarkers, we can integrate into the study the biological questions about drug response on a target through this core. And so basically it's a laundry list of services, laboratory services, that can be plugged into clinical trials as biological endpoints. And this has been very successfully used for a variety of biological questions, from cell biology to molecular biology. So very briefly, I'll give you some examples of the studies that we've done. So we've asked questions about biologicals, and here's a study where we asked whether the combination of IL-2 and IL-12 delivered through a targeting antibody was effective in dogs with melanoma and other tumors. And here is an example of an oral melanoma in a dog disappearing as a result of exposure to these therapeutics. And that's great news that the drug, in fact, made the tumor smaller. But more importantly is we could identify biological surrogates that predicted this happening or that predicted exposure to an effective dose of these drugs. So maybe more important, why did the drug work? Why did it not work? So enrichment could be an outcome of these studies, or selectivization, however you want to refer to it. These biological questions could be answered in these studies, for example, in melanoma. One of the first studies we did through the COTC was to ask whether this targeted approach to deliver TNF alpha actually did what we intended it to do. And we did this by biopsying normal tissue and tumor before and after exposure to the drug. And the drug is in red, and tumor vasculature, which is the tissue to which the drug was targeted, is in green. And you can see that after treatment in the tumor, not in the normal tissue, you see the drug going to the tumor vasculature. So in fact, the drug did what we intended it to do. It went to the tissues that it was supposed to go to, but not to normal tissues, and only after therapy. A difficult thing to ask in a human clinical trial, and you couldn't ask this in normal beagle dogs because they don't have tumor. And in the same way, because they don't have tumor, you'd be misinformed about toxicity in beagle dogs, because the only way that this drug is going to be expressed is when it gets to tumor. So this was a necessary step to ask about the safety of this drug because of these dogs having tumor, and exemplified the point of same species therapeutic index. So in a study that we did with Drs. Trent and others is to sort of take advantage of the heterogeneity that in this example exists between dogs having the same tumors. We could ask what genetic features existed that could allow you to selectivize or identify this dog against the others based on molecular characteristics, and then match them to a set of therapeutics. So the promise of personalized medicine, I think, can be answered by doing studies in dogs. And it would be very difficult, as you can imagine, if it was done in in mice. And then the last example, as Dr. Hellman pointed out, was to look at agents and try to pick the winner when you come down to lead selection. And this is a difficult step using conventional models. But if you have 3 drugs that you can't really distinguish, could you, in the dog with lymphoma, identify a lead using efficacy, biomarkers, and pharmacokinetics? And in the same set of dogs, answer all of those questions. And so that was the point of that study. And I think the answer is that we can generate data that was difficult to generate through other models. So very briefly, repeat the point that one of the important opportunities here is in the same individual dog, you can ask the question of therapeutic index. So rather than saying, does the drug have activity in mouse, and then jumping a wall, is it tolerable in the dog or in the non-human primate? In the same individual, you can ask the question, which I think is an important value of these studies. And everyone likes to see tumors getting smaller. I do too. And it's great when a drug works and you can take a big tumor and make it smaller to the extent that this tumor that was non-resectable can now be taken to a surgical cure. So obviously these are the pictures that everyone wants to see, but the biology that sits behind this is really why you ask the questions, I think, in these dog studies. So I think that there are a variety of places in the conventional drug development path that you can jump in and in a timely manner answer questions to design the best phase 1, phase 2, and phase 3 clinical trial. Just to give you a sense of the intensity of the opportunity, in these dog studies, we can ask PK questions. In tumor, we can ask about the delivery of the drug, as well as the modulation of targets in tumor and surrogate normal tissues. We can collect circulating tumor cells. We can look at a variety of normal tissues to ask about toxicity and therapeutic index. And so over a short, in this case, a long clinical exposure, 6 days, we collect a lot of biological information to identify the best agent to advance in a human clinical trial. So the slide is meant to say that a lot of biology can go into these clinical studies. And as a result, as I said, design the best phase 1 and phase 2 trials. And I think we're now at a point, as alluded to in the questions I asked of Dr. Hellman, to help design these studies, which are really the studies that lead to approval in humans, in dogs. So because these endpoints in phase 2 are so different than the endpoints in phase 3, could we design a phase 3-like trial in dogs to ask about a phase 3-like endpoint in dogs, and then allow you to have the confidence to take those phase 2 winners in humans into phase 3 trials in humans? And this meeting that was convened last year in Bethesda had the simple outcome to say that cancer at the primary site is different than cancer that spreads to different organs, like the lung. So metastasis is different than the primary tumor. The outcome of the meeting said that we should take advantage of the dog with osteosarcoma to ask these questions about metastasis endpoints. And I think that is, certainly an unmet need in human drug development and can be delivered through the dog with osteosarcoma and other solid tumors. So an important opportunity that brought together a variety of interests, but really said that the dog with osteosarcoma can be a critical way to answer these questions. And so there's a mechanism now, in collaboration with the Morris Animal Foundation and others, to take dogs with osteosarcoma, develop drugs for the therapeutic need, which is metastatic progression, ask the question about whether the drugs prevent metastatic progression, and then take those agents and show that they're effective in humans. And when they're effective in humans, they'll become available in a variety of ways for dogs. And so the win-win will certainly be true through the conduct of these studies. And so it's an exciting time for us to have this opportunity delivered. So to briefly discuss some of the perceived risks and concerns, and maybe we'd view these as gaps, and discuss them through the meeting. So it is true that the studies in dogs take longer than mouse models, but if you're strategic and mindful of the value of the question that you're asking, they can be incorporated into the development path so as not to delay the important deliverables in the human development path. And you weigh that against the value of the answer that you're getting. There is patient-to-patient variability, and Dr. Davis is going to be concerned about the fact that these dogs are not all identical beagle dogs held at the same temperature in the same room at the same pH. And that's true, but neither are phase 1 human patients. And I joke with Dr. Davis because we talk about this a lot. But it is true that there is variability, but we embrace that variability because that genetic diversity in a phase 1 population of human patients is the same that you see in dogs, and the variability in how the dogs live their lives is the same as the variability in phase 1 human patients. And Matthew Breen can correct me if I'm wrong, but the SNP frequency amongst dogs across breeds is very similar to the SNP frequency in this room. So dogs as breeds, across breeds, represent that genetic diversity that we embrace in those clinical trials. It is true that if you look at the antiquated view of cancer as something that's diagnosed under a light microscope, that the cancers by histology the dogs get are different by prevalence than the cancers that humans get. And maybe that's a good thing because, for example, dogs get a lot of sarcoma, and that can address an unmet need on the human side. But you're not likely to do as quickly or accrue as quickly dogs with lung cancer into clinical trials as you could in other species. So that is just a point to be mindful as you design those studies. And as alluded to earlier, the target biology may be unique, it may be different, and it needs to be credentialed. So one approach is, as the questions arise, for example, about BRAF, because we have an inhibitor, we can go look for dogs that have mutations in BRAF and prospectively design the molecular model for the disease, or we can use the tissues that exist and start answering the questions in a non-candidate, not example-based method. And maybe that's a need that we need to address, and I think a valuable discussion that can come up in the sessions later on. But sufficient to say the resources and reagents exist to ask those questions, important questions about the biology of these diseases, which we currently can't fully answer. It is true that you need more drug to treat a dog than a mouse. And so again, in the development path, there are times when you'll ask these questions that'll be different than you would if you're asking the questions in a mouse. It is also true that GMP drug is not necessary to do these research studies in dogs. Now, as you get closer to the human clinic, the development team is going to want the drug to look more and more like the human drug, and that is a research question. It's not a regulatory question. The practice of reporting the data and the approach to reporting these data from these research studies, and I make the point that these are research studies, has been addressed, and that, and I think Dr. Vail and others make this important point, the compliance amongst pet owners to participate in the study and to agree to necropsy at the end of the study, for example, is very high and has been consistently shown from these dog studies. There's been oversight guidance given both in the pre-IND and post-IND setting. There is the perceived aversion in biotech and some pharma to rocking the development path by getting answers. But then if you weigh that risk against the cost of finding out late that this didn't work like you thought it is, then maybe it's not such a bad thing to consider. But that perception exists, and I think will come up in later discussions. And I think, again, it becomes a matter of risk and reward. So that's all I have to really say. I'll acknowledge the people who work with me at the NCI in the laboratory and in the Comparative Oncology program, namely Dr. LeBlanc, the director of the program, and Christina Masco, and acknowledge Drs. Wiltrout, Hellman, and to Dr. Barrett for their vision in assisting the development of this program, as well as to all of you who participate in the studies and in the Genomics Consortium. So thank you. I'm happy to take any questions.
Okay, any pressing questions before we start the panel? Well, John, I had a comment and a question. Oh, sorry. Okay, please come to a microphone and identify yourself.
Masa Tamura, Kansas State University. If I understand correctly your report, you said approximately 20 clinical trials completed, only 3 reports made. Is that correct?
I believe that's correct. Yeah, so
Why so small number?
Well, I mean, the progress of the other, so the question is really, so out of the studies that are completed, why does it take so long to publish them? So I think that, the publication strategy often depends on what part of these complex data you want to report on. So for example, we could publish on the first endpoint that's available and have those published immediately, or we could strategically wait for the biology to be completed and then publish them as a set. So these studies take, for example, many months to accrue and then complete, and then to collect the biological data so that you can understand why there's a lag. There's certainly not an attempt to delay publication, and part of the goal was to make them publicly available through +1 through open access. So that process is always ongoing, and it's a matter of what data do you want to include in which paper. Sure. And Christina, is that right? Is it 3 that are published? Oh, so I'm wrong, it's 5. But same answer, I think. But not as bad as I thought. Tanya.
So I had a question about a recent publication from Hay et al. It's in Nature Biotechnology, where he identifies across about 300 different industry-wide analyses that there's an increase in safety failure from phase 3 to new medical products, which is really unusual. It goes from 3% or 9% to 30%. And the understanding is unclear of why that occurs. And we had a lot of discussion around whether it's poor safety monitoring, does it change from phase 3? But we wonder if it's around pharmacovigilance, where they just monitor, track, and then drop patients versus understanding and trying to come up with a new way to monitor and push them through those phase 3 to the new marketing molecular entities. And I wonder if that's a role that's been discussed for the clinical trial consortia, since we really have good tools in our dogs and we've run our tox studies, and if they predict the human toxicities, then you have a good chance to go back.
Well, it hasn't been discussed. I think it's a great point. I think another value that integrates to the value of that point is that we do treat these dogs for much longer periods of time, and they have the syndrome of cancer,
Right.
which changes how drugs are distributed,
Right.
how exposures occur, and probably how drugs are tolerated over longer periods of time. So I think there is that opportunity. Also, you can imagine in the development path, that's a low-risk time to do studies at post-approval settings.
Yeah, I think actually for our safety perspective, our clinicians have a lot of hesitancy in using dogs in clinical trials because they're afraid to generate safety signals or new safety signals in patients. And so if the consortia would actually interact with the regulatory agencies and get some consensus, I think it would help pharma move forward with some of these questions as well. Because often when we interact with regulatory, it looks biased. So it's helpful to have a third party do that interaction and come up with an unbiased decision from the regulatory bodies, and that would help us with our clinicians.
Right. No, I think it's a great point, and certainly I hope that we could get that clarity through this discussion. So we'd be very happy to have that discussion. I don't know, Dr. Layden, you want to? I can't see you, so if you're there, it's right here. So do you want to make any comment, or?
Why don't we go ahead and start the panel discussion, and then we can continue with these questions. So,
Okay. I'll talk. Okay.
Yep, Bev, Sean.
So the answer will be there tomorrow.